I guess we will start. After the walk we do, I would like to welcome you to our EOHSI seminar series. We are delighted to have one of our own presenting today. We have Dr. Cody Smith. He's currently a postdoc at the School of Pharmacy, the Department of Pharmacology and Toxicology. I don't see Debbie here. His mentor is Professor Laskin. He's a core toxicologist, PhD from University of Florida on environmental toxicology. His current research, here's Debbie. His current research is focusing primarily on ozone and other toxicants, but I guess today you're going to talk to us about the pulmonary lipids, inflammation, and ozone toxicity. Welcome to our seminar series, and the floor is yours.
Well, thank you for that introduction. Thank you to the search committee for inviting me here today. Again, I'm Cody Smith. I think I know a good number of people in the room, but the overarching, I guess, theme of my research is understanding mechanisms underlying environmental lung disease. And as I sort of focus on where I want to take my research program as I transition to independence, I plan to more hone in on different signaling mechanisms that might underlie sex-based differences that are seen in environmentally induced lung diseases. And so hopefully you get a flavor for my progression and how I got there throughout the talk. And so yes, today I'll be focusing on my work investigating mechanisms contributing to ozone-induced lung toxicity. So what is ozone? Everyone here is probably familiar with ozone. They've heard about ozone and the ozone layer, which is great because ozone in the stratosphere protects us from UV radiation. But ground level ozone down here in the troposphere is a highly reactive urban air pollutant. It's generated through a photochemical reaction between combustion byproducts such as vehicle emissions and factory emissions and nitrogen oxides in the atmosphere. And so this ozone is so highly reactive and such a cause for concern, it's designated as a criteria air pollutant and thus regulated under the US EPA Clean Air Act. And it's suspected that ozone exposure to ozone air pollution costs the United States upwards of $5 billion in hospital visits and missed school and activity days. So to underscore what a widespread public health concern exposure to ozone is, here I'm showing you a screen grab from the US EPA's website. It's showing, of course, the United States and all of the counties that exceeded ozone levels above that national ambient air quality standard. So any county colored blue here recorded a high ozone level. And so your eyes might be immediately drawn out west where we see these huge expanses of land colored blue. But you have to remember that most of these are pretty sparsely populated. And so if you look up in the northeastern region where we are, we see also a lot of counties and very densely populated counties. So the risk for exposure is exceedingly high in our area. And you've all probably experienced a high ozone level day if you've ever opened up your iPhone weather app and saw this notification indicating unhealthy air quality for sensitive groups. And so that's telling you basically that levels of ozone or one of those other criteria air pollutants are exceedingly high, and you should probably stay indoors if you have some sort of underlying condition. And so why are we concerned about exposure to ozone? Some of the acute effects of ozone inhalation include increased difficulty of breathing, shortness of breath, as well as coughing and sore or scratchy throat. Also causes inflamed and damaged airways. My text font's different, so this will be fun to see what other changes. Chronic exposure to ozone is associated with an aggravation of lung diseases such as asthma, emphysema, and chronic bronchitis. Also associated with an increased frequency of asthma attacks, increased susceptibility to respiratory infection, as well as increases in the incidence of chronic obstructive pulmonary disease. And so it should be noted that these acute and chronic effects of ozone are characterized by a macrophage-dominant inflammatory response. And so it's been the work of our research group in establishing a key role for these phagocytic cells of the innate immunity in regulating both the acute and the later resolution phases of the inflammatory response to inhaled ozone. And when we think about these macrophages, we have to remember that these comprise very diverse cell populations in the lung These include the resident alveolar macrophages, which are embryonically derived from the fetal yolk sac or the fetal liver. They spend their entire lifespans in the lung and serve really important regulatory roles in maintaining surfactant and lipid homeostasis, as well as serving as immune sentinels that respond to some sort of pathogenic infection or a sterile injury to initiate an inflammatory response that results in the recruitment of other cells from the periphery, including the inflammatory macrophages, which are derived from the bone marrow. And so these guys respond to injury and infection. They express high levels of a cell surface integrin CD11b, which confers that migratory potential. And they are broadly defined as being M1 pro-inflammatory or M2 anti-inflammatory, and likely exist along a spectrum of differentiation between the two. And when we think about the role of these inflammatory macrophages in ozone-induced lung injury, we typically consider it to be an imbalance where the overactivity of the pro-inflammatory M1 macrophages overwhelms the anti-inflammatory and immunosuppressive activities of the anti-inflammatory macrophages. And so what this does is it leads to an increase in cytotoxicity and an increase in tissue injury, as well as a failure to resolve inflammation and a failure to repair the tissue. And so a lot of our work has been trying to understand mechanisms contributing to this imbalance and this overactivation. And so the way that we study this in the lab is using a whole body inhalation mouse model. And so here on the left is a picture of our very sophisticated exposure setup. We have our ozone generator that is on the left. This is essentially a box that has a UV light bulb that we hit pure oxygen with to generate the ozone that is then mixed with filtered air and then piped into the plexiglass exposure chamber in the center of the picture, which houses the mice. We monitor the levels of ozone inside the chamber using our ozone monitor, and we can basically just turn the dial on the ozone generator to modulate the intensity of that UV light bulb to control the ozone levels. And so what we do is we maintain the concentration of ozone at 0.8 parts per million for a period of 3 hours. It should be noted that this does, of course, exceed the national ambient air quality standard, but is required just due to inherent differences in the anatomy and physiology of the mouse lung. And so you can imagine that the amount of ozone that's deposited and the sort of the degree of the response is similar to what would be seen in humans performing moderate exercise while being exposed to something more in that environmentally relevant range. And so after the 3-hour exposure, we euthanize the mice 24 and 72 hours later so that we can capture effects in that acute inflammatory and also the later resolution phase. And so some of the things that we look at are markers of lung injury, inflammation, surfactant homeostasis, and pulmonary function. And so when I joined the lab in 2017 as part of our T32 training grant, spent a lot of time learning how to characterize and quantify that lung injury and inflammation in our mouse model. And so one of the things we do is we collect the bronchoalveolar lavage from the mice at the time of euthanasia. This is essentially a wash of the lungs. And in it, we can quantify different components to get an idea of the degree of that injury and inflammation. And so one of the things we look at is the amount of protein present. And so that's what I'm showing you here in the graph on the left. We just measure the protein content using a simple PCA assay. The white bars here represent our air controls. The light red bars represent bronchoalveolar lavage collected 24 hours after exposure. And the dark red bars are those collected 72 hours after exposure. And so what you can see is that there is a significant increase in the amount of protein present in the bronchoalveolar lavage both 24 and 72 hours after exposure to ozone. And what this is telling us is that there's likely some sort of leakage of serum proteins such as albumin into the alveolar space. And so this is telling us that there's some sort of disruption of that alveolar epithelial barrier, some sort of disruption of the tight junctions, which is consistent and a result of lung injury. And so another thing we quantify is the amount of cells present, and we just do this with a hemocytometer with trypan blue dye exclusion. We see a subtle increase at 24 hours post-exposure and a significant increase in the number of cells at 72 hours post-exposure relative to the air controls. And so this is telling us that there's some sort of accumulation of cells in the bronchoalveolar lavage, which would be consistent with inflammation. We also quantify the amount of phospholipids present using a colorimetric assay. And here we saw a subtle increase at 24 hours post-exposure, and a significant increase in the amount of phospholipids present at 72 hours post-exposure. And so this is telling us that there's some sort of disruption in lung lipid homeostasis. This could be due to disruption of alveolar type 2 cell function. All this to say, exposure to inhalation of ozone disrupts that alveolar epithelial barrier, causes inflammation and dyslipidemia. So another thing we do is look at the effects of ozone on respiratory function. And we do this using a Cyrec FlexEvent instrument, which is shown here in the top right. This is essentially a small animal ventilator that we can hook up live anesthetized mice to. And then the instrument performs various perturbations, which allows to quantify different respiratory parameters. And so the perturbation I'm showing you here is called a pressure volume loop, or a PV loop. This is so just to walk you through it, if we focus on the blue line, basically what this entails is a stepwise increase in the amount of pressure being forced into the lungs, and that's causing a resulting increase in the total volume of the lung. And then at a maximum point, the instrument starts to slowly ramp down the amount of pressure being forced in, and then the elastic recoil properties of the lung actually force the air out, causing the lung volume to diminish. So we can basically look at this bottom curve as the inspiratory curve, or the inflation arm. This top curve would represent the expiratory curve, or the deflation arm. And then we perform this perturbation, this PV loop, at increasing PEEPs, or positive end expiratory pressures. And the purpose of doing this is really just to determine whether or not any effect on inspiration or expiration are a result of atelectasis or alveolar collapse. So you can just see that this curve is started farther along the x-axis, meaning it's starting at a higher pressure. And so if we focus on these expiratory curves, what you can see is that the red curve, which represents our ozone-exposed mice, is sort of shifted to the left and up a little bit relative to our air controls. And so this is telling us that we're affecting the elastic recoil properties of the lung with this exposure to ozone. What you can also see is that that response is not affected by changing the PEEP. So this suggests that ozone is causing reduced pulmonary elastance, and this is not a result of atelectasis. And because we're interested in the role of the inflammatory macrophages in this response to ozone, we also quantify the numbers of both the pro and anti-inflammatory macrophages using techniques in flow cytometry. And so what this entails is basically just recovering the cells from the bronchoalveolar lavage and staining them with a panel of antibodies that recognize different cell surface markers. And then you can look at combinations of expression of different surface markers to get an idea of the identity or the phenotype of that cell. And so here we can see that exposure to ozone results in a significant increase in the numbers of both the pro and the anti-inflammatory macrophages, which would be consistent with what we expect to see with some sort of inflammatory response. So again, confirming that macrophages are in fact accumulating in the lung after ozone exposure. We can also look at whether or not these cells are activated using immunohistochemistry. So again, an antibody-based approach to both localize and semi-quantify the degree of expression of either a pro-inflammatory or an anti-inflammatory macrophage in lung tissue sections. And so here we have lung tissue sections taken from air controls and 24 and 72 hours after exposure to ozone. And here on the top, we're looking at expression of induced full nitric oxide synthase, or INOS. This would be an M1 pro-inflammatory marker. And if you look at these macrophages, which are demarcated by the arrows, we can see minimal expression at baseline of INOS, and a really significant increase in the intensity of that brown staining after exposure to ozone, 24 hours after exposure, and then a return to baseline by 72 hours. Now for mannose receptor 1, this would be an M2 or an anti-inflammatory marker. We also see some expression in our air controls. And I think this would just be consistent with the fact that the resident alveolar macrophages are inherently more anti-inflammatory in M2 and do express mannose receptor. But you can also appreciate that there is an increase in the intensity of that staining after ozone exposure, which tells us that those macrophages we saw to be accumulating in the lung through the flow cytometry are in fact activated. Something we've become more interested in lately is the effects of ozone on macrophage bioenergetics. And so This is really important because there's a lot of literature showing that intracellular metabolism of a cell is really tightly linked to its phenotype. And so to do this, we use a seahorse instrument, which is pictured in the top right. This is essentially an extracellular flux analyzer. It allows us to take real-time measurements of extracellular acidification rates as a readout of glycolytic capacity, and measurements of the oxygen consumption rate, which would give us a readout of oxidative phosphorylation. And so we can do this in live cells in real time. And so basically we expose the mice to air or ozone. We collect the cells from the bronchoalveolar lavage, plate them into 96-well plates, and then we basically use different inhibitors of different components of those pathways to get an idea of whether or not those pathways are affected by the exposure. And so here I'm showing you the effects of ozone on the extracellular acidification rate, again, a readout of glycolytic capacity, and the effects on the oxygen consumption rate, that readout of electron transport chain dependent respiration. And so we can see there's a significant increase in both of these pathways after inhalation of ozone, telling us that this exposure is causing an increase in the metabolic demand of the cells. We can also look at the relationship between these two metabolic pathways using this energy phenotype map. So here on the x-axis is the oxygen consumption rate, the electron transport chain dependent respiration parameter, and on the y-axis is the extracellular acidification rate, that glycolysis parameter. What we can see at baseline is the macrophages recovered 24 and 72 hours after exposure to ozone, represented by the red and green data points, are shifted farther to the right relative to the air controls, which are represented by the blue data points, again highlighting that increase in baseline metabolic demand. And we can see that in order to reach their maximum metabolic demand, there's a greater shift to the north on the y-axis after inhalation of ozone, indicating a greater reliance on glycolysis to reach that enhanced metabolic demand. And something I've become more and more interested in lately is the effects of ozone on lipid handling in the macrophages, since we know that this is a critical role of these cells in the lung. And so here, these are cytospin sections collected from air controls and 24 and 72 hours after exposure to ozone. These cytospins were stained with oil red O, which is a dye that recognizes neutral lipids. And so anytime you see any red color, that represents presence of that intracellular lipid. And what you can sort of see macroscopically is an increase in the number of cells that are containing those lipids. And this is represented in the percent of positive cells, here shown on the right. A significant increase was observed at 24 hours, and still an increase, but not significant at 72 hours, telling us that ozone increases lipid accumulation in the macrophages. And so I've also looked at the expression of genes involved in lipid handling. This was just done by qPCR in the cells recovered from the bronchoalveolar lavage. I looked at the expression of two uptake receptors, CD36 and macrophage scavenger receptor 1. CD36 was significantly reduced in these cells in response to ozone relative to our air controls, whereas macrophage scavenger receptor 1 was actually increased at 72 hours. And then I looked at two efflux transporters, ABCA1 and ABCG1, and their expression was also significantly reduced. And so all this to say is that it seems ozone is affecting the ability of these macrophages to handle lipids. So to summarize what I've told you so far, inhaled ozone acutely causes lung injury, disrupts lung lipid homeostasis, and reduces pulmonary elastance. And this is associated with a phenotypic activation of lung macrophages, enhanced metabolic demand of the lung macrophages, as well as altered expression of genes involved in lipid handling. And so as we start to try to think about what might be driving these effects in these interrelated pathways, we realize that there's really one common denominator, and that's Pargamma, or peroxisome proliferator activated receptor gamma. This is a ligand-dependent nuclear receptor. It binds things such as fatty acids, eicosanoids, and pharmaceuticals such as rosiglitazone to regulate the expression of genes involved in intracellular metabolism, particularly those involved in beta oxidation of fatty acids, genes involved in phenotype, where it seems that Pargamma promotes an anti-inflammatory phenotype, as well as genes involved in lipid processing and handling, particularly CD36, which I just introduced. And so I want to determine whether or not Pargamma itself might be a target or somehow affected by exposure to ozone. So I looked at the expression of the receptor itself by qPCR here on the left, and those cells recovered from the bronchoalveolar lavage. We did see a significant reduction in expression of the gene. And then we confirmed that at the protein level by doing some semi-quantitative scoring of lung tissue sections that were stained with an antibody that recognizes Pargamma. And we found that the percentage of macrophages that were staining intensely, indicating a high expression of Pargamma, was reduced after exposure to ozone, suggesting some sort of reduction in the expression of the protein, and again suggesting that ozone is targeting Pargamma expression. And so another skill that I brought to the laboratory was the ability to analyze transcriptomic data sets. And so this is just an approach to profile all of the genes inside of a cell at once instead of qPCR, which looks at one gene at a time. And what it allows you to do is look for significant differences in functionally related genes, which can give you an idea of pathways that might be either targets for or mediators of any sort of toxicity or response. And so here this is RNA sequencing data from cells collected from the bronchoalveolar lavage from either air controls or collected 24 and 72 hours after exposure to ozone. Here on the left in this heat map, each of the rows would represent a different gene. A red color indicates upregulation and a green color would indicate downregulation. And what you can see is that predominantly these genes seem to be upregulated after ozone. And if we look at the right here, these are volcano plots, just another way to represent the RNA sequencing data. On the x-axis is the log base 2 of the fold change. So the farther you are away from this midpoint, the greater the change in expression. And then the higher you are, the farther north you are on the y-axis, which is the negative log of the adjusted p-value. That's the more significant the change, so the smaller the p-value. And so any genes in these top left or top right quadrants represented by the little dots would be differentially expressed genes. And you can really see at 72 hours, there's just many more differentially expressed genes than at 24 hours. And if we use Ingenuity pathway analysis software to try to predict what upstream regulators might regulate and target a significant number of those genes to be found differentially expressed in our data set, we see a lot of things that we expect. And so here in this graph, this is on the bars represent the negative log of the p-value. So the bigger the bar, the more significant the enrichment. And then the dots, black dots, represent the activation z-score. So this is an attempt by IPA to predict whether or not that upstream regulator is activated, which would be a positive z-score, or inhibited, which would be a negative z-score. And so we see things that we expect, such as TNF and CCR2, things we know to be involved in ozone inflammation. But we also do see significant enrichment of Pargamma, and it is in fact predicted to be inhibited. Again, more support for the fact that Pargamma signaling is a target for ozone. And if we actually look at those downstream gene targets in our data set, which would be represented here on the exterior of these little networks downstream of Pargamma, we can see also that they are predominantly colored green, indicating downregulation. So just more evidence that Pargamma signaling seems to be inhibited in response to ozone. And so all of these data provided the foundation for my F32 postdoctoral fellowship, the hypothesis for which was that inhalation of ozone causes dyslipidemia in the lung and reduced pulmonary function by downregulating Pargamma signaling in alveolar macrophages. And so to test this hypothesis, we assessed the effect of pretreatment with a Pargamma agonist rosiglitazone on lung injury and inflammation caused by ozone. And so for the experimental design, we had 4 groups of mice that were exposed to either air or ozone, again at that 0.8 ppm for 3-hour concentration. Select groups were administered either vehicle control or rosiglitazone by intraperitoneal injection, beginning 24 hours prior to exposure, and then repeated daily until we euthanized the mice 72 hours after exposure to air or ozone. And so here we focused on 72 hours because that's where we saw the most changes in the previous analyses. And so we first looked at effects on pulmonary function, again using the side-right flex event. And so here are the expiratory curves from the pressure volume loop. And you can see that as before, we see that sort of shift in the ozone-exposed mice, represented by the red curve, relative to the air controls represented by the blue curve. Again, suggesting some sort of effect on the elastic recoil properties of the lung. And if we focus on the effect of rosiglitazone, we do see a normalization of that pulmonary elastance indicated by that shift back down in the ozone plus rosiglitazone group. And so these effects on pulmonary elastance could be driven by one of two things predominantly, and that would be remodeling of the tissue after some sort of destruction of the tissue architecture, or surfactant dysfunction. And because this is a pretty acute time point and we don't see any evidence of remodeling in our histology, I suspect that is probably due to some sort of disruption in the function of the pulmonary surfactant. And so to look at that, I quantified or looked at the lung hysteresivity using the FlexiBend, and this is essentially a slope of the inspiratory curve divided by the slope of the expiratory curve, and it's a measure of the viscoelasticity of the lungs, which is dependent on surfactant function. And so here in this graph, the two left bars represent our air controls, two right would represent our ozone-exposed mice. The red bars are those that were administered vehicle control, and the gray bars are those administered rosiglitazone. What you can see is that exposure to ozone did significantly reduce the lung hysteresivity, and that administration of rosiglitazone blunted that reduction, suggesting that rosiglitazone is blunting the ozone-induced reductions in pulmonary elastance by regulating surfactant function. And so to look at the effects on pulmonary surfactant in more detail, we went back to our colorimetric assay to first measure the amount of phospholipids present, since these are a major component and a very integral component of the surfactant. As before, we saw a significant increase in the amount of phospholipids present after exposure to ozone, and we did see a significant reduction after administration of rosiglitazone, suggesting some sort of restoration of lung lipid homeostasis. And then we looked at expression of surfactant protein B, or SPB. This is a hydrophobic surfactant protein whose job it is to facilitate rapid incorporation of those phospholipids into a monolayer on inspiration and then back into large aggregates on expiration. And so really it's this surfactant protein B that is what's providing the ability of the surfactant to dynamically regulate surface tension so that you make it easier to inflate the lungs, but then also prevent alveolar collapse on expiration. And so really any sort of imbalance in the relative amount of the SPB to the phospholipids would affect that ability and lead to an increase in the work of breathing. And so here's a Western blot of the SPB levels in the large aggregate fractions recovered from the bronchoalveolar lavage. And on the right would be the densitometry data determined using image J. And so we can see that there is a significant reduction in the relative amount of SPB to phospholipids after exposure to ozone, and then again a mitigation or a blunting of that reduction with administration of rosiglitazone. And so we wanted to look in more detail at what specific lung species might be affected. And we wanted to do this because really I wanted to know whether or not these changes in overall lipid levels were due to a general increase in all lipid species or a change in just one specific species. And so we did some lipidomics with CI and J to quantify the individual lipid species in our large aggregate fractions. And so here this is just a heat map of the differentially expressed lipids recovered from our air plus vehicle group, the ozone plus vehicle group, and the ozone plus rosiglitazone group. Each of the columns would represent a different mouse, and each of the rows is a different lipid. A blue color would indicate a high lipid level, a red color would indicate a low lipid level. And I think what you can sort of take away immediately is that predominantly these lipids in the ozone plus vehicle group are colored blue, indicating higher levels, and that administration of rosiglitazone restores those lipid levels to something a little more similarly seen as in the air plus vehicle group. And then I wanted to look at the amounts of their effects on the phosphatidylcholine species specifically, and that's because these comprise the greatest percentage of the pulmonary surfactant, and they're also the most important in maintaining the surface activity of the pulmonary surfactant. And so here we quantified their levels by just back calculating based on the ion intensity of a deuterated spike in control. We saw a significant increase in the levels of each of these phosphatidylcholine species after exposure to ozone, and again, a significant reduction in each of them after administration of rosiglitazone, suggesting some sort of restoration of lung lipid homeostasis. But what we don't yet know is if this is due to altered production or altered degradation. And so pulmonary lipid and lung surfactant homeostasis is dependent on two cell types, one being the alveolar epithelial type 2 cell, or the AT2 cell. These cells both produce and recycle spent and damaged pulmonary surfactant. And then the alveolar macrophages, which take up and catabolize and degrade that pulmonary surfactant. So you can imagine that any sort of disruption in the activity of one of these two cell types would then cause an effect on lipid levels and lipid homeostasis. So to first look at effects on type 2 cell function, we quantified the levels of surfactant protein D in the bronchoalveolar lavage. So SPD, this would be a hydrophilic surfactant protein. It's produced and secreted by the type 2 cells as sort of a natural response to injury, and then the SPD actually regulates inflammatory responses. And so here we can see in the Western blot and corresponding densitometry data that exposure to ozone caused a significant increase in the levels of SPD, and that this was not affected by administration of rosiglitazone. And so to me, this is suggesting that rosiglitazone is not affecting the ability of the type 2 cells to respond to an injury and to produce the surfactant. And then we looked at the effects on surfactant protein C. And so SPC is another hydrophobic surfactant protein that, like SPB, is involved in regulating the surface activity of the pulmonary surfactant. And so here we looked at the expression of surfactant protein C in lung histology sections. So anytime you see brown staining, again, this indicates positive expression. And you can see that in our air plus vehicle group, we do see expression in the type 2 cells, which are these cuboidal cells sort of in the corners of the alveoli, which are the empty spaces. And so this is good because it tells us that the type 2 cells are doing their job in producing surfactant. And we also see an increase in the amount of that staining after inhalation of ozone. And so this could be due to either hyperplasia or hypertrophy of those type 2 cells, leading them to just produce more and more surfactant, which is a pretty typical response to lung injury. What you can also appreciate, though, is that administration of rosiglitazone did not significantly affect that increase in the staining intensity. Again, more support for the fact that rosiglitazone is not affecting the ability of the type 2 cells to produce the surfactant. So then, of course, we looked at the effects on the inflammatory macrophages, because we know that these cells are involved in lipid handling and lipid homeostasis. And we did this again by flow cytometry. And so as before, we saw a significant increase in the levels of both the pro and anti-inflammatory macrophages after inhalation of ozone. What you can also see is that administration of rosiglitazone did not affect that increase. So it doesn't seem that rosiglitazone is affecting inflammatory macrophage accumulation or phenotype. But then I looked at effects on lipid scavenger receptor expression again in the cells recovered from the bronchoalveolar lavage. And so here we're looking at CD36 and macrophage scavenger receptor 1. We saw a significant reduction in the expression of CD36 after inhalation of ozone as before. Also saw that administration of rosiglitazone significantly increased that expression, so kind of overcame that deficit caused by the inhalation of ozone. We also saw that expression of macrophage scavenger receptor 1 was again significantly increased, and that increase was not affected by rosiglitazone. And so I think this points to the specificity of the response, since we know CD36 is a target for Pargamma, and macrophage scavenger receptor 1 is not. So we think that rosiglitazone is blunting that ozone-induced reduction in expression by activating Pargamma. And so we wanted to look at this with an orthogonal approach with the help of Dr. Behong and Dr. Joseph doing some RNA scope. This is essentially an immunositu hybridization technique to localize and quantify the expression of mRNA in lung tissue sections, in histological sections. So here, anytime you see a red color, that would indicate positive expression of the mRNA, in this case CD36. And so it just, you might be able to tell, but even here in the ozone-exposed mice, those that were administered rosiglitazone, there appears to be more staining for CD36, suggesting increased expression. And if we hone in on what we think are the macrophages, we can also see an increase in the amount of red staining in the macrophages that were collected from the mice administered rosiglitazone. Again, just suggesting that rosiglitazone is increasing CD36 mRNA expression in the ozone-exposed mice. And so to summarize, what I think is going on is that the alveolar type 2 cells produce both the phospholipids and the pulmonary surfactant, or the surfactant proteins, such as SPB, represented by the red ovals. Those phospholipids and SPB spontaneously organize into these large aggregates, and then the SPB facilitates rapid incorporation of those phospholipids into the monolayer on inspiration, back into the large aggregates on expiration. And then those lipid levels and surfactant protein levels are maintained by the type 2 cells, again, taking up and recycling the spent and damaged surfactant, as well as the alveolar macrophages that also take up and degrade the spent and damaged surfactant. And that this seems to, there's lots of literature showing that this is dependent on Pargamma activity. And what I think is happening with ozone is that you're causing that injury to the epithelium that's leading to hyperplasia and hypertrophy of those type 2 cells, leading to an increase in the production of the surfactant. But what I think is also going on is that ozone is inhibiting the ability of the macrophages to do their very important job in maintaining and clearing the excess lipids. And I think that's due to interferences with Pargamma signaling, since I showed that rosiglitazone seems to restore the ability of the macrophages to clear those excess lipids and return the system to the lung lipid, restore lung lipid homeostasis. And so it still remains to be determined how does ozone actually inhibit Pargamma activity. Well, I've done some work looking at a potential role for microRNAs. These are small non-coding RNAs. I first looked at the potential role for microRNA 155, miR30E, and miR17. These are all known to target Pargamma. And so I just measured their expression by qPCR in cells recovered from the bronchoalveolar lavage. And we saw a significant increase in expression of each of these microRNAs after exposure to ozone, 24 hours after exposure, and a persistent increase in expression of miR30E at 72 hours post-exposure. What's more, some of these upstream regulators that I identified to be significantly enriched in the RNA sequencing data set are actually known to target some of these microRNAs, particularly microRNA 155. And so this represents a potential mechanism whereby inhalation of ozone is activating these upstream regulators to induce expression of microRNA 155, which then might inhibit Pargamma expression. But one thing that also stood out to me as I was going through the RNA sequencing data set was the significant enrichment of estrogen receptor signaling in the canonical pathways analysis and estrogen receptor alpha in the upstream regulators analysis. And so this really brought me back to my graduate work, which was focused on estrogen receptor signaling mechanisms, and reminded me of all the literature suggesting a role for estrogen receptor in regulating macrophage activity and function. And so here I wanted to focus on these top two significantly enriched canonical pathways, oxidative phosphorylation and mitochondrial dysfunction, which we know are important in regulating phenotype of the cells. And I just asked the question of what upstream regulators might be really important in regulating the expression of the genes involved in those pathways. And so it turns out that estrogen receptor alpha actually falls out as sharing a lot of connections to those genes identified to be differentially expressed in the RNA sequencing data set and belonging to these two most significantly enriched canonical pathways, again suggesting a role for estrogen receptor alpha in regulating intracellular metabolism and thus potentially phenotype and inflammation. So just a quick refresher on estrogen signaling. I'm sure I don't have to convince everybody here that it does more than reproduction. In fact, sex differences have been observed in a number of inflammatory lung diseases. However, mechanisms are complex. And so to underscore that complexity is this figure on the right. Estrogens can bind to two nuclear receptors, estrogen receptor alpha and estrogen receptor beta. These can either hetero or homodimerize and then interact with co-regulatory proteins and initiate expression of estrogen-responsive genes from estrogen response elements in their promoters. There's also an estrogen receptor alpha that can be palmitoylated and signal from the cell membrane, as well as a G-protein coupled estrogen receptor that can also, both of which can activate various kinase cascades that can then regulate the activity of the nuclear receptors, but then also regulate expression from distinct promoters. And I will say that it appears from the literature that estrogen receptor alpha is really the key receptor regulating macrophage function and activity. And so to look at this in a little more detail, I have some preliminary data through a collaboration with Dr. Troy Roque. Basically, I collected macrophages from the bronchoalveolar lavage from either wild type or estrogen receptor alpha knockout mice, and then I looked at their intracellular metabolism, again using that seahorse instrument. So what I saw was that the glycolytic capacity of those estrogen receptor alpha knockout macrophages was actually higher than in the wild types, suggesting some sort of pro-inflammatory activation, since we know pro-inflammatory macrophages rely on glycolysis. And then I did some qPCR to look at the expression of two pro-inflammatory markers and two anti-inflammatory markers. Here, expression of IL-1 beta and PTGS2, these are two pro-inflammatory markers. Their expression was increased in the knockouts relative to the wild types. And the expression of the two anti-inflammatory markers, arginase-1 and RET-NLA, were reduced in the knockouts relative to the wild types. And so this suggests that these estrogen receptor alpha knockout macrophages are more pro-inflammatory, suggesting an anti-inflammatory role for estrogen receptor alpha. Again, these were naive macrophages, so the effect on ozone toxicity remains to be determined. And so all these data served as the foundation for my K99R00 Pathway to Independence Award, the proposed mechanism of which is that estrogen activates that membrane-bound estrogen receptor to regulate the activity of Pargamma, again, our old friend, the nuclear receptor that promotes M2 anti-inflammatory activation. This leads to an increase in the fatty acid oxidation, which is associated with M2 macrophage phenotype, and that this promotes the resolution of inflammation and limits tissue injury. And so I think that ozone is somehow mucking up this system and leading to a failure to resolve inflammation and increasing tissue injury. And so some of my immediate future directions as part of this grant are to determine whether ozone alters immune metabolic activation of lung macrophages, investigate the role of estrogen receptor alpha in controlling macrophage intracellular metabolism following ozone, and here I will be focusing on that fatty acid oxidation pathway, as well as elucidate mechanisms by which ozone impairs these pathways and prevents anti-inflammatory activation, here focusing again on the role for this membrane estrogen receptor. And so with that, lots of people I'd like to thank, particularly my mentor, Dr. Deborah Laskin, and her entire research team, as well as my co-mentor, Dr. Andrew Gao, and his research group. Also want to thank the countless high school students, undergrads, and rotation students who've all contributed to this work. When I acknowledge the core labs, metabolomics, and flow cytometry, and of course, the funding that we've been fortunate to receive. And with that, I'd be happy to take any questions.
Thank you, Cody, for the interesting presentation. We'll open the floor for questions for people in the room first, and then we go online. So, any questions, please?
Is there consideration of the interstitial macrophages?
Yeah, those are difficult to study. Certainly involved. I don't even think there's really great methods to look at them by flow cytometry or differentiate them from something recruited. But there's definitely evidence that they are involved in injury and inflammation and resolution of inflammation. It's trickier to look at them, but certainly I think that's going to be part of it.
Are they also subdivided M1, M2?
I think they can be activated for sure, yeah. But yeah.
Yes,
I think Macrophages is in first.
Okay. So would at-risk populations like that are in more ozone-rich environments benefit from taking, say, drugs that are PPA or gamma? Is there a downside in, long-term use?
Yeah, certainly. So yeah, I'm not advocating the use of rosiglitazone with its black box warnings and all of that. Here, really using it as a tool to understand the mechanisms underlying this dysfunction in lung lipid homeostasis. People have tried to target lung lipids mostly by affecting diet and polyunsaturated fatty acids, which are anti-inflammatory, but you take a lot more lipids and you're going to end up with a lot more lipids in the lung, which is also going to cause effects there. So it's really hard, I think, to figure out how best to treat ozone inhalation. But I think at the very least, hopefully lending some more mechanistic support for that association between the exposure and the injury.
Questions?
So flipping this more to a population basis, is there any evidence from gene loss studies or other types of things that there are polymorphisms in these pathways that can affect sensitivity?
So Pargamma, I'm not real sure. There is evidence that I've seen recently for a particular mutation in estrogen receptor alpha and being associated with increased incidence or exacerbations of asthma in females. It was a small study, but I think there's something there. Yeah, Pargamma, I'm not sure.
Okay.
I was just wondering what you've read in the literature regarding sex differences with lung toxicity. Like, what have you seen?
Yeah, I think it's contextual. There's certainly there's a new paper coming out every day that, there's differences between the sexes. Sometimes it seems that the females fare worse, sometimes it's the males. So I think it's contextual. I think it depends on when you're sampling, right? Hormone levels are fluctuating, so I think that's going to affect things. So it's hard to say which direction it always goes because I think it just depends. But there's tons of evidence in the literature for sex differences in inhalation studies in mouse models.
Yes.
Hey, Cody, good job on the talk. Thank you very much. It's always nice to see, and we're all staying in our own little lab and not necessarily know what everybody else is doing. So I appreciate being able to see all of the work that you've done. So 2 questions, kind of A and B, so I can do both if you can choose. But the exposures, I guess, do you think it's limited to ozone? You mentioned ozone is one of 6 different air pollutants that EPA regulates. So I guess A is, do you think it's limited to that, or could these studies be extrapolated out to some of those other options in that 6?
I think they're definitely relevant, these mechanisms, to other exposures. I mean, we've seen similar disruptions of lung lipids in response to our chemical warfare agents that we studied, the mustard vesicants. I think, so I think certainly, yeah. And I think it's important to look at the others. Yeah, I mean, surfactant dysfunction underlies, I think, a lot of things, like even like cigarette exposure in the e-cig, and especially with all the oils that are involved. So yeah, I think certainly these mechanisms are relevant to other exposures.
And then part B, at the beginning you said that these macrophages were embryonic, I can't say that word, but they were derived during fetal and embryonic time periods.
Mm-hmm.
So thinking about generational exposure, we do a lot of maternal exposures and some fetal outcomes. And I know that there's literature out there to identify an increase in asthma in these cases, for example. Do you think that could be a sex-dependent outcome then as well? And do you think these fetus could be sensitized to the exposures?
Yeah, so I think my very nascent knowledge on the subject, but that the male and female lungs develop differently, and so there's different things going on. And so I think certainly exposures during development could affect lung function later on and macrophage function, certainly. I think estrogen receptor alpha regulates GM-CSF signaling, which is important for lung development and macrophage maturation. So yeah, I think there's definitely something to be seen there in those early life exposures, certainly.
What about exposures to endocrine disruptors early in life?
Yeah, I think that's yeah, I have a really hard time with that. Just one, how to study it, but understanding what the exposure to endocrine disruptor does to hormone levels in general, which changes all of that, and then how, is there some sort of compensatory downregulation, and then how does that all, I think, lead to some sort of effect later on. But I think there could be something there. These knockout estrogen receptor mice would be cool to look at with some sort of EDC exposure, except they don't really cycle, so you can't really do any of these studies. The goal would be to generate some sort of macrophage-specific knockout that would then cycle normally, so that you could start getting at some effects of the EDCs and the mechanisms underlying that.
More questions from the audience? I have a couple questions as well. So it would be interesting to look synergistic effects of other known pollutants. For instance, wildfires, we know they generate a lot of particulate matter. So that might be something of interest studying.
I mean, there's
The real
research
world
that just
exposure.
came out looking at carbon black in ozone, of course, from Salik Hussein, showing way more severe responses with the co-exposure than the individual exposure. So, yeah, I think you can't look at these things one by one because they're not existing.
Yeah, exactly. I mean, complex mixture. Carbon black might not be a good surrogate
Right.
for wildfires.
Sure.
It's easy to generate in the lab, but might be worth looking into these real world exposures.
Absolutely.
That's the first thing. The other thing, there is a lot of discussion lately about climate change and all of that, and there is also a link of climate change on everything. And I'm sure there is a link between ozone, especially atmospheric ozone, and stratospheric ozone, and climate change. So how do you see your mechanistic studies being kind of a stepstone in terms of estimating the burden of disease related to climate change, assuming that we have a way to measure the effect of climate change on tropospheric ozone?
Right, yeah, so I think we'll have to reassess our exposure paradigms in the lab to account for the climate change penalty, or whatever it's called. Basically, we've reduced ozone levels since the Clean Air Act, but warming climates promote the generation of more and more ozone. So you're really losing that benefit. And so, if you want to stay environmentally relevant, I think you need to be aware of what, climate change is going to do to those outdoor levels.
And now we can open the floor for questions from the audience. I don't have access to the chat room, so please unmute your mic and identify yourself and ask the question. Okay, I guess there are no questions. Okay,
I have
thank
one
you.
more question. Just on your very first slide, you showed the blue spots where there was ozone. I was just curious, why Arizona?
Oh, God.
Pretty sparsely populated.
Yeah.
Is it all sunlight or?
I really can't answer that question.
Okay.
Yeah, I'm not sure I know.
Just curious. It's
Yeah,
Huh?
Cogen plants,
Is it?
energy plants.
Oh, okay.
Same thing in Wisconsin, it's all around the coal, around Madison.
I have one question.
Yeah, go ahead.
So, I know this is maybe difficult at this point, but given everything you've learned about ozone and the lung and how it impacts health and also the potential sex differences, going further forward, what would be some of your ideas about how you might translate this into human studies, or what you might be interested in looking at from a broader, more environmental public health perspective,
Yeah.
given thought to that?
In my very, ignorant mechanistic toxicology mindset, I think it'd be fun to for example, that mutation in the estrogen receptor that seems to be associated with increased incidence of asthma, looking at how that might relate to a high ozone level day in hospital visits or exacerbation of asthma would be interesting. I think the focus on the developmental exposures here would be really, really neat to look at. There's evidence that ozone does affect things like vasculogenesis during implantation, and I think if you're causing this sort of systemic inflammatory response, you're going to see effects on the placenta and growth. So I think it'd be interesting to look at in some of the cohorts that you have now, associating those developmental effects and inflammatory responses that you are seeing to air pollution would be interesting.
And the other point is that, all the lab exposures are acute, right?
Right.
That doesn't really belong to what happens
Yep.
in a community, for example.
Yeah, so that's certainly a future goal, is to start doing more chronic studies, which, and who knows if this disruption of lipid homeostasis will persist even chronically, but I'm sure there's other things going on after that.
And age.
And age, yeah, absolutely.
Okay, thank you, Cody, again for the